I'm hoping that I'm going to tell you something that's interesting. And of course, I have this very biased view, which is I look at things from my computational lens. And are there any computer scientists in the room? I was anticipating not, but OK, there are. So there's one. Maybe every now and then I'll ask you a question. No, I'm just kidding. But so my goal here is going to be to basically actually just give you a flavor of what is machine learning. This is my expertise. And so just actually, again, to get a sense of who's in the room, like if I picked on someone here, like raise your hand if you would be able to answer that question, like what is machine learning? OK, a handful. No, actually one or two. Great. OK, so I just want to give you a sense of that. And I'm going to, most of this is going to be pretty intuitive. I'll try to make little bits of it concrete that I think will be helpful. And then I'll tell you how we used machine learning to improve guide design, specifically for knockdown experiments. But I think a lot of it is probably useful for more than that, but we haven't sort of gone down that route. And so I can't say very much about that. And please interrupt me if something doesn't make sense or you have a question. I'd rather do that so everybody can kind of stay on board rather than it makes less and less sense the longer I go. So machine learning, actually, during my PhD, one of the big flagship conferences was peaking at around 700 attendees. And when I go now, it actually is capped, like it's sold out at 8,000, like months in advance. So because this field has just like taken off, basically it's now lucrative for companies, and it's become a really central part of Google, Microsoft, Facebook, and all the big tech companies. So like this field has changed a lot, and kind of similar to CRISPR, there's an incredible amount of hype and buzz and ridiculous media coverage. And so it's a little bit funny, in fact, that I'm now working at these two kind of very hyped up areas. But anyway, so people in just the mainstream press now, you're always hearing about artificial intelligence and deep neural networks. And so these are so I would say machine learning is a sub-branch of artificial intelligence, and a deep neural network is sort of an instance of machine learning. And so what really is this thing? So it kind of overlaps sometimes with traditional statistics, but in terms of the machinery, but the goals are very different. But really, the core fundamental concept here is that we're going to sort of posit some model. So maybe think linear regression is a super simple model, and you can expose it to data. It has some parameters, right, the weights, and then we essentially want to fit those weights. And that's the training. That's literally the machine learning. So I'm sorry if that sounds super simple and not godlike, like machine learning and everything working magically, but that's really what it is. And right, and so let me just also give you sort of drive home that point. So we're going to posit some sort of model. And so here I'm giving you the simplest example because I think most people here work with linear regression at some point in their life. And so you can think of this as a predictive model in the sense that if I give it a bunch of examples of y and x, and I learn the parameter beta, then for future examples where I don't have y, but I only have x, I can just compute x times beta, and I get a prediction of y. So that's the sense in which I call this a predictive model. And that's very much how machine learning people tend to think of it, where statisticians are often very focused on what is beta, what are the confidence intervals around beta, and things like this. So that's the sense in which there's a lot of overlap, but the goals are kind of quite different. We want to use real data and make predictions. So here it's going to be predictions about guides, and which guides are effective at cutting and at knockout. Right, and so it has these free parameters, and we call these things that we put in here features. And so in the case of guide design, the question is going to be, what features are we going to put in there that allow us to make these kinds of predictions? And so I'm going to get into that in a little bit, but just as an example to make this concrete, it might be how many GCs are in this 30-mer guide, or guide plus context. Right, and like I said, we're going to give it some data. And so in this case, the data for guide design is going to be data from John Dent or others in the community that's now publicly available, where there are examples of, for example, what the guide was and how effective the knockout was, or what the cutting frequency was, for example. And I'm going to get a bunch of these examples, and then that's going to enable me to somehow find a good beta. And of course, we do sometimes use linear regression, but I'll tell you a little bit more about more sort of complex and richer models that let us do a lot more. And then the goal is going to be to fit this beta in a good way. And I'm not going to do some deep dive on that here, but the one way that you are probably familiar with is just mean squared error. And when you find the beta that minimizes this for your example training data, then you get some estimate of beta. And you hope that on unseen examples, when you do x times beta, it gives you a good prediction. So is that sort of make it somewhat concrete what I mean by a predictive model and how you could view linear regression as a predictive model and how you might use this for guide design? Okay, so obviously I'll tell you a lot more. But linear regression is just sort of the simplest possible example. And so in our work, we actually use some of the time what are called classification or regression trees. And so in contrast to here, where you might have, say, you might have a bunch of these features, right? Like how many GCs were in my guide? And then another feature might be, was there an A in position 3? And you can put in as many as you want, and then you get all of these betas estimated. So it's very simple because in that case, none of these features can interact with each other, right? You just add x times beta 1 plus x times beta 2. So we call this like a linear additive model. In contrast, these trees allow very sort of deep interactions among the features. So this might be how many GCs. So of course, this is not suited to the features I just described, but this might be some feature like, I don't know, proportion of GCs, because now it's fractional. And then this algorithm, which is going to train the betas, so find a good value beta, will sort of through a procedure that I'm not going to go into detail for all these models how it works, but it's going to somehow look at the data and determine that it should first split on the second feature at this value, and then it will sort of keep going down that. It says now partition the examples in my training data like this, and then on the second feature in this way, until you end up at the sort of leaves of this tree, and these leaves are the predictions. And so when you do it for the training data, whichever training examples here end up at this leaf, you basically take their mean, and that's now the prediction for that leaf. And if you take a new example, you basically just pipe it through these sort of rules, and you end up with that kind of a prediction. This is a bit simplified, but I think it's a good conceptual view. And this is just another way of thinking about it, is if you only had two features and you drew them like one against the other, then effectively every time you make a branch here, you're kind of cutting up the space. So that's also just another way to think about it. And so this is all over the press nowadays. And whenever I give these talks, there's a bunch of young, hungry grad students who say, "Did you do deep neural networks?" Because that's what everybody wants to do now. And so deep neural networks are kind of like a really fancy linear regression. So you could think of these as the x's in linear regression. You can think of this as imagine there's only one thing out here. I should have done a different picture. That's just y. And so this again is a mapping where you give it the x's, you do a bunch of stuff, and out you get a y here. Except linear regression, is this very simple thing. And now you can see there's all these other kinds of, we call these like hidden nodes. And so there's this complicated mess now of parameters beta. And again, I'm not going to go into it. I just want to give you a sense that like linear regression is this very simple thing. And there's a lot of other models that let you do much, much better prediction. And these are typically the kind of models that we use because they're more powerful if we care about prediction. But the flip side is they're actually very hard to interpret. And so if you want to ask a question like, was it the GC feature, that is most important in your guide design, which is what I always, get a question like this. And we can do our best to kind of poke and prod at this machine, but it's always a little bit ad hoc. It's hard. The more complicated the model, then the better we might predict, and the less interpretable it is. And so there's always this kind of tension. So right, and so what are some of the challenges? So I've sort of shown you sort of some like increasing amount of complexity in some models. And so one of the big difficulties is if I posit a very complex model with a lot of parameters, then I need a lot of data in order to actually fit those parameters. And if I don't have enough data, what's going to happen is the parameters, you're going to find this very specific setting of the parameters that effectively memorize the training data. And the problem then is you give it a new example that you really care about. You say, I want to knock out that gene, and it's never seen that gene. Because it's sort of memorized, but we say it's like overfit to the data, it doesn't actually generalize well to these unseen examples. So there's another tension here, which is you want kind of complex, rich models that can capture very complicated prediction spaces, but if you don't have enough data to warrant them, then you're going to shoot yourself in the foot. And so when you learn in this area as like an undergrad or grad student how to do rigorous machine learning, this is one of the things you need to understand, how to control that knob, how to know if you have enough data for the model you're looking at, and things like this. And so in this case, this is kind of fun for us to do the guide design because a lot of people are using this now, and we know we didn't super overfit it because people are telling us that it's useful. But we did a lot of due diligence on our end to convince ourselves that that was correct as well. right? And even just asking, how can I evaluate a data set? There's a lot of papers out there where people sort of dabble in machine learning, they go use something and they prove that they have this amazing predictive capacity. And in many cases, because it's such a popular area now, people just kind of jump in and they do something, and these evaluations are actually not rigorous. And so you need to read things very carefully. You can't just look at a result the way you can look at a gel. Maybe I'm wrong here, correct me wrong. If it's not fabricated, then it's not just what you see is what you get. You need to think very deeply about what they've done and if that makes sense. Right, and then of course, like in linear regression, for example, we assume that the error term is, for example, Gaussian distributed. So this is like a very sort of technical, precise thing. Of course, almost in most cases, and every model has some sort of assumption like this. And basically, when you're working with real data, every model is wrong. Like, there's no way. I mean, the real model is physics, right? And we're not using physics-based models. They're hard, they're not scalable, and basically, it's almost unheard of, except in a few select fields of like molecular dynamics. And so again, this is one of sort of the tricks of the trade, is how do you, and to do it rigorously, is how do you know like that this assumption, even though it may be violated, that your model is still okay? And so that's another consideration. Sometimes we think it's okay, but maybe if we can violate it less by doing something different with the data before we give it to the model, maybe we can do better. And then in a lot of the work I do, sometimes exactly a very good model, but it's just really, really slow. And so you need to be clever about actually how to speed things up. And it might be slow when you're trying to infer those parameters during the training phase. So I'm going to often refer to like a training phase where you have this data, and then there's sort of a deployment or test phase where you're just using the model and making predictions, but you don't know the right answer unless you're evaluating. So that's sort of, I guess, just a sort of overall little bit of a view of a slice of machine learning. Are there any questions so far? Okay.
So one of the challenges, of course, is you have to have a handle on all the features that are important.
Yeah, absolutely. And so in a way, one of the sort of miracle breakthroughs of deep neural networks, and that that's touted, is that you don't need to know them, because it sort of is such a rich machinery. It can create features on the spot. But the problem with them is it needs a vast amount of data to do that. And the other problem is they've really only been shown to be state of the art. They've broken like 30-year benchmarks in on things like speech processing and vision tasks, which are images and speech, which have particular properties like continuity in time for speech, continuity in space for vision. And people are applying them everywhere now. But in many cases, people say, I used a deep neural network, but you know what? If they just used some simpler model, they would have done just as well. But that is sort of, so what you said is exactly right. And when you don't use deep neural networks, and even then sometimes if you don't have a lot of data, then yeah, this sort of feature engineering is what they call it, is what do we put in? Do we give it just the GC content? Do we give it this? But kind of one of the beauties of machine learning is that you can posit a whole bunch of things, throw them all in, and if you have enough data, then it can tease apart what is relevant and what's not. But again, there's always this tension of like, if you give it too many things and you don't have enough data to warrant it, then you're going to get a bit of nonsense out.
But is it true that you still can't look under the hood and see which features are important?
This is a very active area of research, although I would say it was a bit obscure up until very recently. And the reason it's become much more important is because deep neural networks are so important, and people are using them for problems that physicians are looking at a CT scan and stuff, and in medical areas especially, people want to know, well, why did it make that decision? Like, I'm not going to give my patient this or that or not do that because your weird, crazy deep neural network just popped out 0.8, And so this is a very active area of research, and there are things you can do, but it's I would say it's always, in some sense, an approximation over what, because at the end of the day, it's something it's like, a human works as a whole, right? And then you ask some very specific question about some individual thing. Well, like in biology, we ignore the whole rest of the system just to try to understand because we don't have a choice, right? And it's very much like that in machine learning as well. So you do ask and you get something that's useful out, but you know that by ignoring every other system and every other thing that you can only go so far. All right, and so I just want to give you also a little bit of a flavor of sort of what I would call the two main branches of machine learning, and everything I've just told you about is what we call supervised learning. And so in supervised learning, why is it supervised? It's because in the case of like guide design that I'm going to tell you about, the way we do it, the way we build this predictive model is I give it a supervisory signal in the sense that I give it a bunch of guides, and for each guide I know how effective the knockout was. And that's the supervisory signal. And so if without that, I mean, how, in a sense, how could you make predictions, right? You can't build a predictive model if you have no examples of how well it's worked in the world. And the trick is to take some limited number of examples and do something that generalizes to a much larger set of data. And so that's and this is, obviously used for any number of problems in biology, outside biology, in finance, like anywhere in chemistry, you name it, this exists. And so my own background, I should say, is been a little circuitous. Like my undergrad was actually in physics, and then I actually did computer vision, and then I got into computational biology during my PhD, and I've meandered around like through sort of immunoinformatics, proteomics, and so in fact these are kind of problems that I have looked at, and now more recently I'm doing some of this CRISPR guide design. And so, right, and one distinction also is, am I predicting a yes, no, like knockdown or knockout versus not? That's classification, because you have a class of yes, it was versus no, it wasn't. And regression is sort of like, how much was it knocked out, or like, what fraction of cells, or what was the cutting frequency, something that's not discrete. And we don't need to pay a lot of attention to that, but in our papers on this topic, we have shown that people like to view this problem as 0, 1, like it, cut or it didn't cut. But if the assays that you guys produce like give much more information than that, and it's kind of crazy to just ignore that. And therefore, we want to be in a regression setting where we're actually predicting real values. And mostly we care about the rank, because the kind of scenarios that we've been trying to tackle are, someone says, I want to knock out this gene. So we want to go through, and I should say everything we've done here is also for Cas9, just like the wild type Cas9. And so we want to, we say, our goal is to essentially say, rank all the possible guides for that gene for Cas9, and then compute the on target according to our models, compute the off target, and depending on your use case, you balance those in some way that you think is appropriate. And so we want rankings here. Like, we don't care if we know the exact number, although I guess some of our users say, like, well, what does that number mean? And that's very hard to do because of the nature of the data that we take in and the sort of different assays that get incorporated, and how we have to kind of massage the data to throw them all together, which I'm not going to talk about here, but is a very difficult problem, actually. Right, and so in this setting, it's pretty easy to evaluate the model. It's not trivial, but it's much easier than the second setting, which is unsupervised, which I'll tell you about in a second. And the reason it's easy is because I have some set of examples for which I know the answer, right? And so the kinds of things that we do is we basically fraction off the data. We keep some amount of our data away from us, and we just never look at it until the very end. And then we do a bunch of our own sort of methods on this other part, and we are allowed to look at it as many times as we want. And at the end of the day, then we go kind of once to that final validation set and say, how well did we do? And we can do that for different methods, right? So we can look at competing methods from the literature or competing methods we make up and say, like, okay, well, is this, how do they fare? And so in that sense, it's like that there can be problems with that, but it's much easier than in this case. And so this is unsupervised learning, and I'm not going to talk anything about that other than this one slide here today, but I just wanted to, if you've heard this, thought it might be useful. So probably everyone here at some point in their life was doing something with gene expression, and you would draw these little plots from, I think it was from Berkeley, right? Wasn't it Eisen, Mike Eisen? Okay, maybe this is a different, I don't know, who here has drawn these plots some point in their life? Okay, at least more than people who could define machine learning. So, right, so one axis here are, I can't actually see, what is it? Yeah, so each of these would be a microarray experiment, every column here. And then people would say cluster these for, people with colon cancer or something. And then they'd say, oh, are there subtypes that we can see by clustering them according to this tree? And you would sort of cut off the tree and give you some clusters. So in this case, the sort of analog in guide design would be, I give you just the guides, but I don't tell you anything else. Like, I don't tell you how efficient efficient the knockout was at all. And then so you're just trying to understand some properties of this set of guides. And for guide design, this, as far as I know, this is not interesting or useful. I don't know what you would do with it, but in other cases it is. And I've spent a lot of my life actually in statistical genetics, where you can do some really cool stuff. So when you're doing GWAS, like genome-wide association study, and you're scanning through looking for each marker, is it sort of related to this phenotype or this trait? One of the things that really messes you up there is when you have heterogeneity of, say, populations from the world. And so one thing people do is they kind of do this unsupervised learning. In this case, it's called principal components analysis. You can take all of the genetics and you do this process, and then say each person in this room, like say we've measured you at 23andMe, whatever, you have all your genetics, and then you can do this unsupervised PCA, and now each person will get 2 numbers out of this analysis, this unsupervised analysis, and you can plot them here. And when you do that, it actually kind of recapitulates the map of Europe if you happen to know where they're from. So that's not going into the algorithm. And so that's kind of cool. I mean, I haven't told you exactly why we want to do this, but it's just kind of a super neat result and totally irrelevant to this talk. Right, so that's my sort of very quick and rough intuitive intro to machine learning. And I'm going to jump into how we use machine learning for the on and off target problem in guide design for knockout. But again, are there any questions at this point? Okay. All right, so this one, the first one I'm going to talk about is on target. And that one's already published. This is with John and other folks at the Broad. And then this one is in revision now, although it's on bioRxiv, not the most recent version. And of course, our overall goal was to have some sort of end-to-end guide design service, but we got to do this in pieces. There's almost no end to this, right? Even when we have our current one, there's going to be a million things we can do. And I think usually, so I actually often talk more to computer scientists, rarely to biologists, and I have to explain in great detail what it is we're actually doing. And I suspect everybody in this room knows, but just to be clear, again, I'm going to focus on the knockout, although often the assays and the modeling we do are actually cutting frequency. So in this case, I'm considering knocking out this gene. I'm going to use Cas9, and maybe there's like NGGs at these 4 positions. Of course, it's more than 4. And the idea is I want the one that's going to most effectively knock it out. Or I want to rank order them in some way, right? And then let the user decide what to do. And so I want the best guide and to guide me to the good position. And then, of course, the flip side is if I've chosen that particular guide, then I want to make sure I'm not disrupting the rest of the genome. And so we model these separately. And now I'll go through one at a time. Right, so this is a little schematic of what I mean, which I've been alluding to, I think, in the introduction about how we're going to do guide design. So again, this is going to be supervised in the sense that John's going to give us a bunch of examples in his wet lab where he's measured the knockout efficiency. And what I'm going to do is I'm going to take the guide, and it's actually, we have a 30mer here, so it's 23 plus I think it's 4 extra on one side and 3 on the other. And to be honest, like that's not something we ever played with much. Somehow I think we just started using 30. Maybe we tried just the guide and this helped. I don't actually remember right now, but it's a 30mer sequence. And then we're going to learn some function. And so again, in the simplest case, this function might be linear regression, where you're effectively just learning some betas. And then it's going to allow us to make predictions. And the game is going to be to collect as much data as we can and to fit these in a suitable way. To pick a good model and then to fit it in an appropriate manner. So that's the setup. And right, and so these, I've just drawn here kind of 3 of the key components, the decisions you have to make when you're doing this kind of thing. So the first one, and which Dana just was mentioning, is like, what features do we give it here? Like somehow I go from this, ACTG guide to some numbers, and I'll tell you a little bit about that. And then the other thing is we need some measure of the supervisory signal as how effective it was. And so that's not, as a computer scientist, that's not in my control, but John and you guys are super clever and managed to figure out how to measure these things. And then our job is to think about, when you measured it for that gene and that gene, are those comparable? And can we throw them together or not, and things like this? Or should we, transform it in some way so that it better adheres to the model that we're trying to use, maybe Gaussian noise and things like this. And then finally, what model are we using? Or are we going to try a bunch of models, and how are we going to decide which one is best? So right, and so John, in this first paper, he had some data already there, and then he gave us some more, and overall we had, I think I have a slide on this, it was for just 15 genes. He did systematically every possible place you could deploy Cas9. Okay, great. So right, he had this one he'd already published, which was they were all cell surface proteins, and so you could use flow cytometry and fluorescence to separate them and get some measure of knockout efficiency. And then the second class of genes was using a drug resistance assay, where I guess it's known, and I guess they show in the paper as well, they reestablished that when you apply this drug, and you successfully knock out the gene, then the cell survives, and otherwise not. And so using these kinds of tricks, and probably many more that you guys develop, we can get some sense of if the protein was not there, not functioning. And you can see here, I guess, large data is in the eye of the beholder. So for modern day machine learning, this is like minuscule, like it's a little sneeze basically. And that makes machine learning really hard, actually. It makes it easy because things are fast and they're not unwieldy, and I don't need to worry about memory and compute time, but it's really bad for getting a good model. And that's actually one of the big challenges in this area, is there's very limited data at the moment. And I'll never forget one talk I saw years ago by Google, like before machine learning was super famous, and they actually, at one of the big machine learning conferences, they did this plot, and they basically showed the performance as you change through these different models. And you see this model's better, this and this. And then they went to the next slide, and they said, okay, now we just made the data set 100 times bigger. And basically, that's so dominated the difference between the models that you're like, just get more data and don't worry about what model. But when you're in this regime, you should care about the model. And to be fair, even in that regime, that was just one specific problem, but I think it kind of drives home a very nice point. And so, right, I mentioned that different genes and different assays like kind of yield a measurement in a different space. And I'm not going to. We have ongoing work as a sort of machine learning project on how to handle that in a rigorous, nice way. And in the meantime, we're doing some stuff like changing it to ranks and normalizing with the gene and stuff. If you want to know, we can go into this offline, but I'm not going to talk about that here. Right, and so now back to this sort of featureization, what we call featureization of a guide. So right, we have the 20-mer guide, and then a 3-mer after the PAM, and 4 before, and we need to convert that 30-mer nucleotides into something that's numeric. Because any model I want to use in machine learning, fundamentally it assumes there are numbers there. They can be real value, they can be negative, they can be discrete, it doesn't matter, but they gotta be numbers. And so, we're not the first people to have this problem. There's a rich history of computational biology and ways to do this. And so this, like we did, this is a pretty standard thing, but just to help you wrap your head around some of the ways in which we might want to do it, imagine that, so we're going to, a lot of the features we're going to use are based on the nucleotide content. And so what we're going to do is we're going to kind of make a dictionary where if there are 4 possibilities for a letter, then each of those basically gets, we call it one hot, because one of these bits is on and the rest are off, right? So this is the one that's hot. That's the one that's hot there. It's called a one hot encoding, but I think you can see you just enumerate however many things you have, and then the code for it becomes however many there were. So here there's 4, which means there's 4 digits, and then you just kind of randomly give a 1 to A in that position, to T in this. And now what you do is when you want to convert this to a long numeric thing, first you look up the T. So you start with a T. So then you get 0010. Those are the first 4 numbers in the long numeric string. And then you look at the second letter, and it's a G. So then that's going to be followed by 1000, and you keep going like this. So that's going to give you some very long string, but we're not going to stop there. We're going to actually also do this for what we call order 2 nucleotides, and we get this sort of other kind of dictionary. And you can do this up to order 3. You can do all kinds of other stuff that I'm not going to go into. I just want you to like sort of wrap around like in your head, how can you actually do this? And also the way I've just described this to you, like in this example, it's position specific in the sense that I start here on the left and I ask, was there a T? So this refers to the first position. And when I do this for every guide, it always refers to the first position, right? So when this is important in the model, it's because it's important in the first position. But I can also do things that are sort of position independent. So, and I'm going to actually do that not just for GC. I'm going to do it for every 2 pairs of letters. And I'm just going to say how many GCs were there, how many TGs were there, and things like this. And I'm just going to tack those on to this thing. This thing's just going to get longer and longer and longer, right? And this is where we need to be careful. Like, is it so long that I don't have enough data to actually deal with it properly? But in this case, like, even though they're really long, because so many things are zeros, it's actually not quite as long as we think it is in terms of the amount of data needed. And then something that comes out as important, and so this is, we sometimes call John our like Oracle featureizer, because John is, 1, he's smart, 2, he has very good intuition, and 3, he's up on a lot of literature. And so he said, there's crystallography experiments that show that, something happens at the 5-mer proximal to the PAM in sort of a cohesive way, followed by something over here and over here. So maybe when you're doing the thermodynamics, in addition to looking at the whole 30-mer, maybe also look at it just in these subportions and things like this. And so we interact with John and we get features in this way as well. So we kind of use our rich history of just computational biology, and then we use domain-specific expertise. And if we had sort of an infinite amount of data, we could probably skip thinking through these things at all. Right, and so the last part, like I've now told you a little bit about how we do this, and you guys know more about this than I do. And so now the sort of last puzzle piece is what kind of model are we going to use? And so I've already told you a little bit about these regression trees. And so actually a lot of our CRISPR, these are just very, they're very rich models because you can get these interactions, right? If I split on this feature, let's say this is GC content, and then I go down to here, and this is, let's say, A in position 1, then this is a very strong interaction between these 2 features because I have to go down this path before I get it. And so you can get these very complicated interactions, which is also why it's hard to interpret, because you kind of need to like look at every interaction all at the same time to kind of understand it. So what we actually do is we actually use a bunch of those models together. There's a way to combine these models to do even better still. And this is called boosting. Does it have the name on here, boosting somewhere? Boosting. So boosting is a way where you take some sort of somewhat simple model. These aren't simple models, but we actually cripple them a bit. We keep them very, very simple. And then we apply this boosting technique. And so let me just explain to you intuitively what boosting is like. This is, I think, pretty easy to understand on an intuitive level. You take your data, and you fit the parameters on your training data. And then you go to your training data and you ask, how well did this fitted model do on every one of my training examples? And then you weight each of your data points inversely to how well it did. In other words, if on that training point, after I fit the model, it did badly, then it's going to get upweighted. And the stuff that it did really well on is going to kind of fall out. It's going to get a very low weight. And then you reapply this learning algorithm, you refit another tree, and you add the results, then you start averaging the results from a sequence of these trees. And so there's a whole theory underpinning why you can do this, what that's actually doing. But on an intuitive level, that's all it's doing. And so it's, in some sense, it's refocusing its energy on getting those things that it didn't get right by subsequently adding in another tree. And these turns out to be very powerful models in a large number of domains. And so, in fact, that is what we use here.
So you also do an iteration where you drop out a feature and see whether it made a difference? A couple of features, because you can simplify that.
Yeah, so I guess there's 2 things we do when we're doing things. One is we try to get the best model possible that we think will be the best, like for whoever wants to use it, we really think this is the best thing. And then another thing we do is we want to interpret it as best as we can. And so sometimes we drop out a feature because we think maybe it'll be better. But usually dropping one feature is not going to make a difference, because if it's just one feature and we drop it out, it would have known it wasn't useful anyway, usually. Like it could be that if you posited that there were just like millions of features and then you got rid of 500,000, that might help you if they were irrelevant. But if it's just one irrelevant one, it can figure it out, or else it's just doing a bad job anyway, basically. But then you might do it to, like, we have some figures in our paper about here's the accuracy with all the features we finally used, and here's the accuracy if we pull this one out, to give people a sense of like, well, how important was it? But that doesn't necessarily really tell you, actually. So it's actually more nuanced than that, because it could be that if I use this feature by itself, and we see this all the time. I think this happened with like microhomology, which we don't put in our model. If you put that in just by itself, it turns out to be quite or somewhat predictive. I don't remember how predictive this was. But if you take the model we've already settled on and you add it in there, it makes no difference, because things are, like, for example, nucleotide features cover GC content, and GC content is a proxy to thermodynamics, and you don't know what's a proxy for what and things like this. And so because everything's interacting, like, all you know is in the context of this model, if I pull it out, it doesn't change the performance. But that doesn't mean that it's actually not predictive, right? And then the flip side is, if I put it in by itself and it's not predictive, it doesn't mean it's not predictive, because I could have put it in with a bunch of other stuff and together it might have been predictive. So this is why it's actually hard to do that, and it's also why it's hard to interpret. Yeah, but we do what we can in that vein to sort of poke and prod as best as we can.
You said something on sort of the previous slide, and I wanted to make sure
Yeah.
got it right, which is that sort of, we're having to go in because the data set is small and say, these are the features we hypothesize are important, and sort of give it that a priori. And we might be leaving things out.
Absolutely.
To understand the system.
Yeah, completely.
So it seems like what you were saying was that if we had a larger data set, we wouldn't have to be as correct about those features going in, because the model could start to find some of those more complicated things on its own?
So that, yes and no. Yes, the larger the data, the more complicated a model you can give it with more parameters. And the more complicated a model it is, then with infinite data, it can figure out these features. But so you need to both increase the data and increase the complexity of the model. So if you did just one or the other, then that's not enough. Unless you're in a setting where you're using too complex of a model, then you should be, and then you increase the amount of data. But yeah, I think your intuition's correct. I just wanted to make sure you know that these things need to go kind of in lockstep, in a sense.
So getting more data is not a magical fix for us not understanding which features are important.
No, and in a sense, the more data you have, you can afford to do more complex modeling, and the more complex modeling, the harder it is to tease apart what's going on, unless you went to some physics-based thing, but like nobody really does that. Right, and so at the end of the day, we're using these boosted regression trees, and so when this is for the on-target problem using those 15 genes, and the colors look slightly different. Okay, so anyway, and John and colleagues, actually, this is the paper they had in revision when I first went up to John and started talking to him. And that was the state of the art for on-target predictive accuracy of knockout. And that's performances in blue. And so this is flow cytometry data, this is drug resistance, and this is combined. And this is when you train on one, test on the other. Of course, we're careful. When we do that, we don't literally train on that whole data set and test on it again. We partition it in this way that I described. This is just a shorthand notation to say we're only considering this data when we do that. And similarly here. And then you can see the boosted regression tree is doing better. And actually, also, everybody had actually been doing classification, and we can show through a series of what we call experiments, like in silico experiments, that it really, you don't want to do classification here. You want to do regression, because the model can use that fine-grained assay information. Right, and so some of the features that come out here, and I've put them into groups. So, for example, the topmost, and again, take this with a grain of salt, because the other thing is with these models, sometimes you can fit them this way. You could just tweak the data. You could imagine taking out 5% of the data. You could refit it, and you might get something very different here, because there's many ways in which you can get an equally good predictive model. And so, as much as people really want to latch onto these lists, and we make them, like and whenever we send something in for review, they're always like, well, we need to understand this better, but there's really only so much you can do with this kind of an analysis. But I think it gives you a flavor of stuff that's not important will not be in this list that much, and I think so there is something to be had from this. Just shouldn't read it too literally. But so the position-dependent nucleotide features of order 2. So one example of that would be there's a TA in position 3. And this is the importance of all of those features together. And it turns out in this case to be the most important thing. And then position-dependent order 1. So that's like, is there a T in position 21 or 20 or something like this? And all of those together turn out to be important. And then position-independent order 2, this is something that includes GC, right? Because one example of a position-independent order 2 feature is how many GCs were in that 30-mer. But you can see even with the GC content in there, the thermodynamic model that we use becomes out to be also important. And moreover, those little bits that John inferred were important based on crystallography, like actually taking subsets of the 30mer, turn out also to be important. So you can see that's the 16 to 20, 8 to 15. And then one thing that for knockout is important is actually where is the guide like actually cutting, right? So if it cuts at the very end of the gene, it might still form a functional protein that passes this assay kind of thing, whereas if it is the very beginning, it's sort of less likely. That's not, you know, if you look at the data, that's not crystal clear, but it's there enough that it comes out to be important. And then it turns out the interaction, again, this is what we call another John Oracle feature, which is between the nucleotides on either side of the GG and the PAM. If you actually say, I want to know what both of those are at the same time, that turns out to be important. And actually, and GC count still comes out as well. And then you can drill down, right? These were like groups of features. These were all the position dependent order 2 features. And then this is just the top of the list in rank order of the actual individual features. You know, if you go to the SI, you can find all of them. But so percent peptide is again just a measure of like, were you 50% through the peptide where you targeted it, or were you 100% through? So you can quantify that as a percentage or as an absolute number, like I was 30 nucleotides in, and we put both of those in. Into the model. The other one's amino acid cut position. So those actually are very important. And then the second most important one is actually the 5mer and the thermodynamics. Right, so, and this is actually, it seems, I don't know if anyone's here using it, but a lot of people tell me they're using it, and there are actually 2 startups that are using this now as well. And everything I've just told you is actually from this paper, but we're right now in the process of bringing in more data to try and improve it and trying to see how chromatin accessibility and other things like this might improve the model or not. But that's in the works. That's not out yet. And then one thing, I actually don't usually put this in my talks, but I thought I'd get a question about it. Is just like, you know, what if you had more data? How much better would you do? And this is like one of the first things John asked us, and he said, maybe you can draw a plot, something like this. So, you know, we actually had, I guess we had 17 genes. And so we always want to partition the data when we're testing, right? So if I have 17 genes and I want to use as much of it as possible, I could use 16 to train and test on 1. And then I could keep doing that. I could shuffle and then do it with a different one. And so that's this end here, and that's the performance there. And you can sort of see as I let it use fewer and fewer number of genes to train, the performance kind of goes down. And John was like, you know, is it going to plateau? Does that mean we have enough data and getting more data won't help? Which, you know, of course he was hoping we would say yes to that. And you could kind of make up that story in your head based on this plot, but there's a whole bunch of caveats with that, and that it kind of looks like that here, but I don't think it's the case. And if you want to know more, you maybe ask me after, but yeah.
What's the Spearman R?
Oh, I'm sorry. Yeah, Spearman R, I'm going to talk a lot about. So when we want to evaluate how well it does, let's imagine that I have 10 guides that I've held out, and I've trained my model, and I say, how well does the model work? So I know the knockout efficiency for those 10 guides, and I have my predictive model, and what I want to do is basically take the correlation between those. I don't know if we care about the exact values, but we care about the rank order, and so we do it actually with the ranks. So we just convert the predictions into ranks, and then we convert the real assay values into ranks, and then we do like a Pearson correlation. So it's really just getting at, are my predictions in the right order according to the ground truth? And Spearman is doing it on the ranks because there's a lot of messiness with the raw values that we get out here. And this, I think, is enough that it is still very useful.
And is 1 perfect then?
Yes.
5 half perfect?
Yes, that is correct. Yeah. Mm-hmm. Exactly. So that's the on target. And now I'm going to what time? I'm all confused. I thought we were starting at 8, but it started at 8:30. So okay, I think this is good. Yeah, lots of time. Yeah. So now I'm going to talk about the off target. But unless there are questions about the on target first, that's sort of the much simpler one from a machine learning perspective. It was much easier to do. The next one was a bit of a nightmare.
Can we come back to it later?
Yeah, we can absolutely. Yeah.
The parameters that had the largest effect.
Yeah, definitely. Yep. Okay. Right. So this is a much more challenging problem for us for a number of reasons. And so one thing is that we're now kind of looking for accidents, right? We don't have this perfect complementarity between the guide and the target, which you have for on target. We're saying, well, did it accidentally do something there? So now we need to consider mismatches between the guide and the target. And moreover, we need to do that across the whole genome. So now suddenly, instead of just saying, does this guide and this target, is it going to cut and knock out the gene? I need to scan the entire genome. And then I need to somehow predict how active is it going to be at each of those places in the presence of some number of mismatches. So this is just like actually wildly more difficult and computationally expensive. And so, I mean, one is I have to scan the whole genome. So that by itself is really annoying. And then from a modeling perspective, the fact that I have to tolerate mismatches means, and I guess in computer science lingo, we'd say this is like a combinatorial explosion of possibilities. Like, and I'll show you how it explodes. Is that the next slide? No. But like, so the more mismatches I tolerate, kind of the more examples I need for the model, because there's just so many possibilities. And there's actually, I think there was even less data here than for the on-target, depending how you count data, but it's really, really limited. Right, and so this very simple 3 steps here. It took us a while even just to get to that. And so ultimately, though, this is how we think of the problem, is given a particular guide, our goal is going to be to find out where in the genome there are likely to be off-target activity using our model. And then we also want to summarize it into a single score for that guide. Because people don't want to, like, for a single guide, they don't want to like ponder. I mean, they might for some use cases, they might want to do that, but I think a lot of people, depending again on the use case, they want just a single number with the option to drill down about where the off-target activity is. So what we're going to do is they're going to start with a single guide, and then we're going to scan along the genome and filter essentially like to a short list of potential active off-target sites. And then once we have that short list, then we're going to use machine learning to score each of them with one of these predictive models. And then we're going to aggregate them all into a single score. And so most of the work that we did, well, all the machine learning is down here. And one could, in principle, actually use machine learning up here as well, but there are just already too many moving parts. And so we actually just stuck around to doing a heuristic, which is to basically cut it off roughly, actually, it's not at 3 mismatches. Huh. I just realized these slides, it's something a little bit different. I think it actually goes up to 6, but it allowed to ignore the 3 distal or something like this. So I apologize, I don't have exactly the right details here. But this shows you what I was talking about, a combinatorial explosion, which is if I tolerate only one mismatch, so this is a back of the envelope calculation, right? Like, let's say there's 100 sites genome-wide roughly, and the more I go, like the more sites I have to consider. And so when I do this filtering down to a short list, like I can only go so far. And that's also why actually it stands to reason the machine learning could maybe do better here, because this is a very rough criterion of just like number of mismatches. And we know that that's not what's driving the model because we've done the modeling, but it definitely has a big impact. And so it's not a bad thing to do. Right, and there's lots of software out there that actually does this, like tons of things that will go and find all of these things for you. And I guess our add-on contribution is really to do the machine learning on top of that in a way that is really better than what's out there. Right, so that's all I'm going to tell you about this first step. It's basically just a heuristic where we say we allow up to some number of mismatches. Now that's our short list for this guide. So right now the problem is how do we score the off-target activity for each member of that short list? And so there have been some previous approaches on this from your community that's putting out these great papers, and they're making very clever models that actually do surprisingly well in a lot of cases, given that they're like just sort of like declared by fiat to be the model. It's really amazing. And so we thought, let's bring in machine learning and see if we can do any better here. And at the time we started to do the CFD, which actually was John's model, in the paper we wrote together, but we ourselves didn't get to the off-target problem because we were just didn't have time for that paper, is basically counting frequency of knockdown according to some very simple features. And I'll tell you a little bit, I think I have a little bit more about that. And so we're going to, again, do this machine learning thing. And so the last picture I showed you like this was for on target. And in that picture, it did not show you the target. If you might recall, it had only this one column, which was the guide, and then it mapped directly to the activity using just the guide. And so, but now, because of the mismatch, I have to include the target as well, right? In on-target, by design, these were complementary, and so this was redundant information. But now, there might be mismatches between these, so if I know this, I don't necessarily know that, so I have to throw that into the mix. And so now this function I want to learn is a function of the guide and the target. And then I'm going to play the same game. I'm going to get as much data as I can, and I'm going to try and learn this function. And actually, in this case, we're using only 23 mer, not 30. And the reason for that is I just think the data sets we had 23, and it was a real pain to make it 30. So we just left it at 23. So these are how you store your data in your Excel sheets may impact how we do machine learning if we get a bit lazy. Right, and so now I need to also play this game again where I go from these now, the guide and the target, and go to some vector. And mostly I'm going to do the same thing I did for on-target. So everything I already told you, I'm going to do that for the guide again, and that's going to go in here. But then I need to somehow also encode the mismatch. And I can do that in different ways. I could, for example, I could just say there is a mismatch at position 8, but not tell it what it is. And then separately say one of the mismatches was a T to an A. Or I could say there was a mismatch at position 8 and it was a T to A. And so there's some flexibility there, and we actually do several of those things. And again, if we had enough data, we wouldn't actually have to enumerate these different things. We could just kind of give it the components, and the model could figure out what the features should be, but we have very limited data. And then this is where it also gets super complicated. So again, I mentioned this sort of combinatorial explosion of possible number of mismatches. So like, I don't know how much data there is that has multiple mismatches, but it's like, maybe 1,000 examples. And you saw how many mismatch examples there were, right? Like, I mean, thousands and thousands. So we've almost no data, I would say. It's almost like no data. However, what John did is he built, and this is kind of going to sound insane, I think it is insane, but it actually works, is he took just CD33 and he enumerated every single mismatch guide target pair, and he measured the knockout efficiency for that. And from that we can, like if it's just one mismatch, we can build just a one mismatch model and not consider the combinatorial explosion. And that gets us a one mismatch model. And that's actually what we do, is we build this one mismatch model just from CD33 data. And I'm super curious to know if we actually had some more genes in there, how the performance would change, since in general, we're not at all testing on CD33 here. And then the thing is that we want to be able to take any arbitrary example and kind of bootstrap ourselves using that single mismatch model to get a prediction for it. So how am I going to do that? What I'm going to do in the first, on this slide so far, is I'm going to pretend that this 2 mismatch, or it could be 3, if there were 3 mismatches, there'd be another figure here. I'm going to break it down into examples, like pseudo examples of single mismatch guide target pairs, even though that's not what it was. And I'm just going to do this because I don't really have a choice, because there's just not enough data. And then I'm going to assume that they are, for example, independent. And if they're independent events, I can multiply them together and I can get a score. But they're not really independent, and it's not really the right model. So we're actually going to use the very limited amount of multi-mismatch data with a really simple model that has very few parameters to actually learn to do something better than multiplying them together. And that buys us a little bit extra. Doesn't buy us a lot, because there's just very little data, so there's not a lot we can learn. And if we had more data, we could probably do much, much better here. Does that make sense? How I take CD33 single mismatch data, I build just a single mismatch model, which we hope is not specific to CD33, and it's clearly not, because we do get some generalization performance. But to be clear, off-target prediction is actually not great. It's pretty low. And then we break down examples into the single mismatch, and we could, in principle, just multiply them together, and that would actually not do that badly, but we add on a layer of machine learning with a very limited amount of multi-mismatch data, which we get mostly from guide-seq data. And we're doing all unbiased assays here for off-target, because we don't want to focus on kind of weird things. So we're doing unbiased assays like guide-seq that search over the whole genome, even if they're not as sensitive. And again, right, so we actually use these boosted regression trees for the single mismatch model. And then this is, I said we have to use a very, very simple model here because we have so few examples. And here we just use linear regression. And actually, you may not know what L1 penalized is, but it's basically a super constrained version of linear regression where you kind of force or coerce the parameters to be closer to 0 than they would otherwise. Yeah?
For the previous slide, does it take into consideration the abundant affinities of the mismatches? For example, like here you have a TC
Yeah.
match, whereas a GC match.
Right. So it doesn't do it in the sense that we don't compute binding affinities of those things, but we give the model that information of that it was a T to a G. And so if it has enough examples of that to tease apart that was useful, then it could, in principle, learn that kind of a thing. Yeah.
And this doesn't take into account the nearest neighbors of the mismatch.
So actually, in this layer here where I said we do something that, so one could multiply these together, but instead we learn a model here. In that model, some of the other models out there were not machine learning, take into account how close together the mismatches were and things like this. I think that's what you're maybe asking, or
Actually,
no.
So if a TC mismatch is in a
Yeah.
TTT context
Yeah.
on each side,
Yeah.
it'll have a different influence
Yes, yeah.
on stability than if it's
Right,
based on both
right.
sides.
So we implicitly have the ability to capture that, and the way we have the ability is that when we encode this mismatch, one of the ways in which we encode it is we say there's a T to C mismatch in position, I don't know what that is, 7. And then separately, we have features that say there's a T to C in position 7. And so it can combine these things and learn, it can put the pieces, like the Lego pieces together to infer that potentially if there's enough data. But we don't do it more explicitly than what I just described.
Because that was one of your features, the nearest neighbor identity was one of your features in the on-target model, right?
In what sense? Like what, so what did the feature say?
Well, just that you go down the sequence and you say, not just that there's G in position 1, but there's GG in position 1 and 2.
Right.
And that was one of the features.
Yeah, no, you're right. Actually, we could augment the way we have featurized the mismatch specifically to more directly to encode that, and it might help. In principle, if we had enough data, it wouldn't help, but we don't have a lot of data, so it might help. And so that is something we could try, actually. That's a good idea. Yeah. Right, and so now we actually have more, so this is in revision, we're hoping to submit in the next few weeks, and we actually, maybe somebody in this room said, please get some more data and really validate this. And so we started working with, I'll mention at the end, because I don't know how to say Ben's last name, and Keith Young at MassGen, and so we have some more data and show that this generalizes beyond what I'm going to show you here, but I don't have that in the talk yet. And so, right, there's the CD33 single mismatch data. You can see there's about 1,000 guides. And then this is sort of, I think, from the original GuideSeq paper, and there's 9 guides up to 6 mismatches. And so it says there's only 354 which are active in any way. That is that the GuideSeq assay finds them at all. And then, so if you just look at N total, you think you have this huge sample size, but those are basically zeros, like non-active targets you get by searching the genome up with this short list, like say up to whatever 3 or 6 mismatches. And so that's your kind of candidate list. And then GuideSeq says, well, I found activity at these very few. So even though this N is huge, this is tiny, and that is really important. So we don't have, in a sense, we don't really have anywhere near that number. And then, so there's a nice review paper from Max Hausler and colleagues where they were comparing on-target and off-target algorithms, and they did a nice compilation of some other unbiased assays that included GuideSeq, and so we use as a separate data set basically all of Hausler minus this GuideSeq, and so those are kind of 2 different assays, and those were all cutting frequency assays. I don't remember the details, but again, you can see here that once we subtract out the GuideSeq, this is tiny. I mean, this is just really, really tiny, and it's actually CD33 is the only really big data set we have, and it's kind of remarkable we can get anything at all out of this. Right. And so one question that, with the on target, there's a whole bunch of standard measures we evaluate things like the Spearman. You said, what is the Spearman? And that's sort of just something that you just think immediately you do, and you'll see this in all kinds of papers in many domains. And if you're doing classification, there's something called the ROC curve, which we're not doing classification, so I don't tell you, but all that is to say, there's like standard things that people just do like this. And Nicola and I spend like literally months trying to figure out how to properly evaluate this, because one of the problems is there's a really inherent asymmetry here, right? Like say, especially if you think of therapeutics, but in any case, if I have a predictive model and I have a guide target region and it's active there, like it's going to be active and it's going to disrupt things, and my model says that it's not active, that's the kind of mistake it is, then that's a really bad mistake, right? I'm going to use that guide and something bad's going to happen, like someone's wet lab experiment's going to mess up, or I'm going to kill someone. And on the flip side, if my model, if there's some region, some target region that it's not active, but my model makes a mistake and says it is active, well, that kind of sucks, because I'm not going to use a guide that might have been a good guide, but there's not some sort of devastating consequence, right? And so there's this inherent asymmetry, and we wanted to take that into account when we were evaluating things, because we want to evaluate in a way that's most meaningful for what people care about. And so we ended up, we're aware of this asymmetry, but we didn't also, but where in this sort of asymmetry space do we lie? How do we kind of weight those two extremes? And so in the end, this is what we decided to do. We used a regular Spearman correlation here, and this plot, it just turns out to be easier to visualize this way. This is a relative plot where this is an improvement over CC top, which is sort of a older model out there. So it's the baseline, and then you can see basically from the Broad how well they do. And let me explain what this knob is in a second. But so this is just Spearman R. Oh, this got moved around that block. Well, whatever, it's fine. And now what happens is on the other extreme end, over here, what is this? It's also a Spearman, but it's a weighted Spearman. So now each guide target pair is weighted before I compute the Spearman. And in particular, it's weighted by the activity. So if a guide target pair is not active, it falls out altogether. And that's because of this asymmetry. So that's the most extreme case of the asymmetry I just described. This is, if you're not active, I don't even care if I get you wrong. And what you can do is you can turn a knob that smoothly varies from one end to the other. And that's what this is. It's a little complicated. The details I won't go into, but you can see it's smoothly varying. It's not doing something crazy. And so what's really nice is there's no theoretical reason for this, but almost invariably when we draw these plots, you find that one model systematically lies above another model. And that's nice because it means that no matter where you are in this regime, you should use like the one that falls on top of the other one that dominates it. And because it's actually a bit hard to conceptually understand where you are in this space. Like we designed this and I find it a bit hard to understand as well, but at the same time, I think it's a very useful metric. And so you can see in this case, when we train, we always train on CD33 and we never test on it. It's always the basis of the single mismatch model. And CD33 doesn't appear anywhere in any of the other data, as far as I know. And then we also add in the guide seq, which has the multiple mismatches to do the second level. And then we test on the Hausler et al. That does not include guide seq. And that's the relative performance of the different models. Does the figure make sense in the motivation, even if it's maybe not the kind of thing you're used to seeing? We're not used to seeing it either. And then you can flip it around. Where now, again, we always use the CD33, but now we use the Hausler combined data set, and we test on the guide seq, and you see a similar kind of pattern. And again, we can drill down a little bit. And this is actually just from the CD33 single mismatch model. Oh, yeah. Yeah.
Just thinking about this in the previous slide. Trying to think, does that have a 50-50 chance of getting this right?
Yeah.
Either it looks like guide seq helps Hausler better than Hausler helps guide seq.
Yeah, it's a little bit hard to. So let's see. That looks like that. Remember, these are improvements over CC tops, and actually in the preprint and in the paper, it tells you the actual correlations, which are pretty low. I forget now, but I think they're maybe around 10 or 20%. I don't don't hold me to that. But the difficulty is that the number of active guides in each of these two is very different. The GuideSeq has about 3 times as many active guides, so in some sense, it has a better chance of building a good model, and so you have to be careful about things like that.
That's an issue for you all the way along, is what's the quality of the data?
Absolutely.
Comparability
Right.
of the data.
Definitely. And so one of the decisions we have to make is like when we're trying to evaluate how well things do, we want to flip things around as many ways as possible to show the underlying sort of modeling approaches are solid, right? That if you just tweak this or that, you still get this, which is why we do both of these. But at the end of the day, we need to say, we're going to deploy a model. Do we train it on just the GuideSeq? Do we train it on the GuideSeq and the Hausler? And, and so this is partly like just an intuition we have by looking at things, and we talk to John, and in the end, like the final deployed model is just trained on GuideSeq, actually. Plus the CD33 right now. But again, we're trying to get a better one going as well. But yeah, absolutely. And I think in the on-target paper, we actually have like a matrix where it says if you test on FC or train on the flow cytometry, test on the drug resistance, how well does that generalize? And like we try to always do these kinds of things. And Hausler in his review paper has a super nice, he did a very expansive set of comparisons where he, across many data sets where he does this kind of thing. If you take their model, but you test on their data and all this kind of thing. So there's kind of no end to this. So you do as much as you can to think that, that what you've done is solid.
I guess in the previous slide, I'm just a little confused why you lose the difference as you move towards the weighted true activity in the 10 -3.
So actually, we've since again these should be updated. I literally flew here last night after giving a talk at UCLA yesterday, and I haven't had a chance to update it. But in fact, one thing I'm glossing over here is what we realized actually just during the current revision is that as you turn this knob over this way, the effective sample size goes down. So for example, over here, I basically turned off anything that's not active. And so those are falling out of the computation. And a consequence of that is that this is a very high variance. Like that estimate is not super stable. So we now actually compute effective sample sizes and truncate. And I'm pretty sure on our new plots that this, it actually gets truncated something like here. I have to go look. I apologize that I don't have it. But and so all I can say is I don't know specifically why it takes these shapes. What I do know is that as you go more and more to the left, the effective sample size goes down, and we should consider that, which we hadn't up until now. Right, and so this is just sort of groups of features and how important they were. And so this here, unsurprisingly, is there's a feature that says you had a T to A in position 3. This is mutation identity plus position. So that everything together, you tell it the position and the identity and what it went from and what it went to. So that's actually super, super important in the model. And then if you tell it just the mutation identity, but you don't tell it the position, so you just say it was a T to an A, but I don't know where, then that also turns out to be helpful. And if you tell it just the mutation position, you say, I don't know what the mutation was, but it was in position 3, then that's also helpful. And then this is translation versus transversion. Is that what it's called? Transversion versus transition. Yeah, you can tell me what that is, what we put it in. I guess that's, what is it, like a C to A is a what?
C to T is transition,
okay.
A to G is transition,
okay.
Everything else is a transversion.
All right, so I guess we read somewhere that this might help. But what's interesting is this sort of drives home a point I was mentioning earlier is for every example, if I tell it the mutation identity and the mutation position, but I tell it sort of as 2 separate features, then these regression trees, in principle, they can combine those 2 things and make up this feature. So if I had a lot of data, I wouldn't have to give it this feature, but through experimentation, we can see we don't have enough data that it doesn't actually do that, and so that's why we give it all 3 of these, and they all turn out to be important. And so this is I don't know, for me as a computational person, this is interesting. I don't know if it will be for you, but this is an example of like we wanted to show that kind of different components of what we've thrown into the mix here help one on top of the other, and that we're not doing things for no reason. And again, just for visualization purposes relative to CC-top. So I've mentioned that, so red is this final model we deployed, which is the one I've described to you. And what happens though if instead of, remember I said there's a second layer model where instead of just multiplying together the single mismatch things, I add in this extra machine learning. If I don't add in that extra machine learning, then I end up on this blue curve. And so you can see that extra level is helping. And probably if we had a lot more data, it would help a lot more. And now what the one below it is, it says, what if we do the same thing as what I just described, the blue curve, but instead we actually use just the implicit features from CFD. So CFD was what John and guys had did. And basically, they don't pitch their model as a machine learning model, but as a machine learning person, you can look at it and you can say, aha, the features they used, it turns out they used one feature, and the feature they used is just, there was a T to A in position 8. That's the feature that implicitly they're using. If you use just that feature, you actually do pretty well, right? Like that's actually driving a lot of the signal. But again, you buy more by adding in these other features. And now finally, if instead of doing regression, where I'm predicting like ranks or like real values, I actually just say, was it active versus not? So I pick some threshold, and this is actually what most other people do, then you get actually terrible performance. And this shows you that you're throwing away a lot of information. Right, so now so far what I've showed you is I'm gonna pick a guide, I'm gonna filter using up to some number of mismatches to get a short list. And then for every potential off target in the short list, I've now shown you how we've trained a model and how it fares compared to other models that do similar things. And now I wanna tell you how can we aggregate this into a single number. And this is a kind of complicated in the sense that it depends on your use case and all kinds of things. But I think having something is better than nothing, and people can ignore it if they want, but people seem to want it. And so how do we take all the scores that result for this one guide from step 2 and aggregate them into something meaningful? And other people have done this also, but not with machine learning. And so what we're going to do is the following. We're going to take this one guide. We're going to scan the genome for the potential off targets. So, this, I can't remember this. Maybe this is like 3, 4, 5,000. I'm not sure anymore. For each of those, we're going to apply the model I just told you about, which takes the single mismatches and combines them with this M function into one number for that guide target, as sort of something like that probability of activity, let's say. And I do this for a whole list of targets. Of course, if I did a different guide, I would have a different list and a different length of list probably as well. And now I can think of this as having some distribution of off-target activity. As well as how many there are, right? And so now I can say, well, can I do machine learning on that? But now the question is, what is the supervisory signal here, right? Like, so before it was guide seek and things like this. So what's my aggregate measure? And so again, this is not, John said you should try this viability data and the non-essential genes. And so the idea is that if I target a non-essential gene, so my desired on target is in the non-essential gene, then if I have no off targets, then the cell will survive and it'll be viable. And to the extent that there are off targets, it may not be viable. And so the question is, why is it not viable? So the reviewers have complained a little bit and said, well, really what that assay is capturing is it's not capturing some sort of sum total effect of off-target activity. It's basically capturing, was one of your off-targets essential or not? And then the reviewer said there's actually so few essential genes that basically like this is meaningless because like how can you get anything? And so it's true that if you hit an essential gene as an off-target, it will die. But it's also true that there are so few of those that this is not what's driving it. And there's now actually 3 different papers out. Did I add them the citations? There's 3 papers out that show, probably by people in this room, I don't know, that sort of the more cuts you get, the more likely the cell is to die. And we now have experiments that show that is, we have experiments that show it's not knocking out essential genes that's driving this experiment. Like we can show that because we can actually insert that information in and show that it doesn't help any. And that's not surprising because there are actually so few. I think it covers like 2% or less of the genome sort of by sequence. Right, and so we use that as our supervisory signal, and then we again play this game where we partition the data, we train, and then I say how well does it do. But again, there's very, very little data to do that, and so we use again a very simple model, which is it's based on linear regression. And when you do that, oh yeah, right. So so we do this with 2 data sets of Vana and Gecko. And again, just on the non-essential genes. And I forgot to say, so we have this distribution. So what kind of features do we use from this distribution? So I think we have sort of the number of things in it. And then we somehow will compute things like what's the average, what are the quartiles, and then we might say, what are the average in just the genic off-targets, the average score from our model, and things like this. So it's incorporating what's genic versus not, and we can put in the essentiality there, which is not helping. And then something also the reviewers have asked for is like, what if you, in this whole off-target thing, you incorporate the chromatin accessibility? And so that turns out to be very difficult. It actually does help, but it's really hard to get the right cell type. And so even though it helps, we're not right now putting it in our model, just because for almost all the data we have, we can't even get chromatin accessibility data. So in principle, that would be nice, but it's a bit too early to do that, I would say. Right, and so when you do this, and now you can actually, so on this data set in particular, it's pretty low, this final aggregation score, but it's certainly it's actually highly significant. And you can say that this is our aggregation model. And I didn't say this explicitly, but to the extent we do well on aggregating, it means that we also did well on the individual score guide target scoring, because if we did wrong on that, then you would have no hope of doing the aggregation. So aggregation is some sort of summary measure, in a sense, of how well we did on those, plus how well we combine them together. And if you did either of those things wrong, so if the previous model I told you about was just like bogus, you would get nothing out of this. And then CFD is actually John's way of combining stuff. It shouldn't say website, it's not even available on the website, it's just in the paper. And then this is from the MIT website. And then you can flip it around again. So this was, right, train on Havana, test on Gecko, and then you can flip it around and you see, actually, right now the correlations go up a lot more, so you get stuff more around 0.15, and you see a similar ordering of things.
How long till MIT takes the website down?
I don't know. Is anyone in the room from that group? No? Okay, I'm curious to hear if yeah, we should talk to them. Right, and so we want to put this all together now, and so this is very cumbersome. So if you look at the available online tools for off-target activity, the way they work now is you request some information, say about some gene, and then you wait like 5-10 minutes, and it like emails you something, and it's sort of it was annoying for us to do evaluations. Like we had to get someone to program something that would ping it, wouldn't ping it too much, and would wait, and then stuff would time out, and like these kinds of things. And we wanted something where people could just go, and it was actually pre-computed. And because I work at Microsoft, we have a lot of computational resources, and so it's kind of fun sometimes to make use of them. So we decided we wanted to and actually when we first told John we were going to do this, he was like, that's impossible, you can't do that. And so we said we want to pre-populate, for starters now it's just the human genome, for every coding region, compute for Cas9 every possible on-target and off-target score, as well as be able to drill down into all those off-target scores. And so we actually, for the first revision, we populated that, although it wasn't for the first submission, and we're now tweaking it. But that takes roughly, we run for about 3 weeks straight on 17,000 cores. So which probably no one here could do that with wherever you are. And so it's kind of, sometimes it's fun to be at Microsoft to do that kind of stuff. And so we're building this site, and we've tweaked it now a bit based on the revision, but basically there's gonna be different ways you can search, but based on gene, based on transcript, and then it'll search through anything that satisfies Cas9, and then you can like click on the guide and drill down into the off-target activity, and we'll give you some sort of basic information like was this off-target in a gene or not, but we'll also give you the position and chromosome in case you want to like look up other things. Yeah, so that's unfortunately not live right now because one, we've been modifying things and we're now repopulating it, and then once the revision's in, like hopefully, and this is full, then hopefully very soon, maybe in the next few months, I hope this is publicly available. And if in the meantime you want to play with it. I have to talk to my collaborators. We might be able to give some people private access. Yeah, sorry.
The score is big thing.
Oh, you know what's really funny is, yeah, the funny thing is, I think that I have this, after we submitted it and it came back, we realized we had something flipped around, and now I can't remember if, like, on this example, what is what anymore, and this will be made clear when it's released, and I don't...
It never is on a lot of sites.
I see.
Oh, great, a number.
Oh, yeah, so this has little tooltips, like if you hover above the question marks, you get information, and one of them will tell you that, and I just don't remember, and I know we had it wrong, not in the sense that anything in the paper was wrong, just that like something on the website was like the wrong direction. Yeah, and so, right, and none of this would be possible without a wonderful set of collaborators, and very much Nicolo, who's my computational collaborator, who has an office next door to me, and John at the Broad, who's just been wonderful, and the three of us have a bunch of projects on the go right now. And very recently, we hooked up with Ben and Keith to generate some more GuideSeq data to validate these off-target models. And Michael Weinstein, who I think he actually just left UCLA, he actually did all the coding infrastructure for basically that whole search. If you give me a gene, like find, or a transcript, like find all the possible regions on the genome up to this number of mismatches, and then he pipes that into our machine learning. And so those two things work together. And then some folks at Microsoft who help with the website and the population of the database and all of these things. So I think, yeah, that's it. Thank you. Yeah, we haven't yet looked at, other CRISPR. The question is, like, how well does it work for other CRISPR? How well does it work for other organisms? We don't know yet. Other than in Hausler's review, he did something interesting where he basically said our model was state of the art as long as it was, he compared like a whole bunch of organisms like mouse, zebrafish, and all of these things. And the zebrafish we did really badly on, but he could show that it's not necessarily, it's unlikely to be because of the organism, and it's more likely the difference between in vitro and in vivo. And so actually we now have privately retrained in vitro models, and he's taking a look at those. So, and I don't know even almost what that means. Someone told me the biochemists and some other community don't even use these words in the same way. And so that like someone will yell at you depending how you say them. So all I know is that like that's a dominating factor over organism. And that there seems to be, like, this is just me, like, I can't rigorously point to a picture, but sort of my memory from all the things I've seen is there's pretty decent generalization between organisms, but I'm sure that if you have data for a particular organism that, probably that will help.
I think one of your limitations is the only information you have to develop your features is the sequence information.
Yeah.
And you mentioned chromatin.
Yeah.
To the extent that nucleosome location, compaction has an influence on any particular target, that's completely outside.
Yeah, absolutely. Yeah.
There are data out there, but they may be more cell-specific
I see.
Than you'd like to have.
In fact, when Ben and Keith were about to generate this new data, like we decided together like how to do this, and and I said, can you do any of these cells that have chromatin? Because none of our guide target data has chromatin data. We ended up having to do it with the viability data at the aggregation step, and he said like basically no, like not because he was unfriendly and uncooperative, just for whatever restrictions they had. Like he even then, as we were generating new data and wanted to, we couldn't there. But if you guys know where there's good data for us to use, or you're generating some and then it's out at some point, feel free to let us know. But yeah, this is a big amount of data, and then these kinds of auxiliary data sets both stand to really improve these models a lot.
That's sort of the issue of you may have some features that you could discard, the machine could discard them, but you also have some features you don't have access to. Absolutely, undoubtedly, yeah. Would you mind going back to some of the on-target?
Yeah.
Just one thing that struck me, the importance of melting temperature in nucleotides 16 to 20 is more important in this histogram than the melting temperature at 8 to 15, 3 to 7. And if I remember correctly, so is 16 to 20 next to the PAM, or is it?
Yes, it is.
Those are the ones next to the
Yeah.
PAM. So that makes sense.
Okay.
Okay.
Good.
So, I mean, one of the things, I grew up as a nucleic acid biochemist, and one of the things I would like to know is what physics features are being reflected here, even to the extent the nucleotide sequence is predictive. How can we understand this in terms of thermodynamics, interaction between the DNA RNA hybrid and the Cas9 protein? You know, what atomic level features are reflected?
Right, and I think it is possible to go in that direction. It's just that you don't get it for free by doing what we did. It could also be that by doing that, in fact, we somehow improve what we did because we have limited data, and maybe like in the thermodynamics. In principle, if we use a rich enough model, it should just compute the thermodynamics if it's necessary, right? But it's obviously helpful for it that we've told it the thermodynamics. So if there are other physics-based things, we could go out of our way to test to see how important they are with all the caveats I've mentioned. Or we can add them in and say, does it also help the model? So if there are things that are particularly interesting to the community that we are kind of readily computable, we can throw them in.
Seems like an obvious thing to look at. We were looking at the crystal structure 2 days ago, and it looks like it goes to this intermediate conformation state where it's testing whether its guide already matches the DNA. And it's kind of doing this. So once we understand a little bit more about what's happening.
I feel like that's how, I may be wrong, but I think that's why it got broken up this way, is that there was this sort of one phase where it was like feeling things out, and then if it was okay, it would sort of keep going to the more and more, but that's not my area, but I have a vague memory of that.
When you consider the sequence, I think because for the single guide RNA scaffold, if you have different sequence of the scaffold, I think since some paper that the scaffold can form a secondary structure with the spacer sequence. So when you make this model, are you relying on single scaffold sequence or?
What can you tell me what the scaffold is?
It's like a user RNA.
I see. And is that, this is now getting at the limit of knowledge of the system, and that's not constant across all these experiments?
Oh, I think people now, most of the experiment is still using similar
I see.
structure.
I see.
Now people are making
I see.
variations.
Okay, so probably it's the case that all the scaffolding was constant here, and therefore we didn't model it. But if people were changing the scaffolding, then this could, and presumably it is important, which is why people are changing it, then absolutely we could put that into the model. Yeah.
I think one thing she's getting at is that the target spacer sequence that you guys are changing can mess up folding of the rest of the RNA. So if it complements with different parts of the RNA, so that might be a.
But still, if you're not, even if that's the case, if in the data I have I'm not changing that, then I still don't need to put it in there. It should be able to just figure that out. Because if something's not changing, it's not informative, by definition. Yeah.